we're good to go.
Okay.
Okay, our speaker tonight is Dr. David Meyer. He got his bachelor's in geology at the University of Michigan and PhD at Yale. He is retired from the faculty on the geology department at the University of Cincinnati, where he taught invertebrate paleontology, paleoecology, geology and biology of coral reefs, historical geology, and dinosaurs. His research interests focus on fossil and living crinoids, coral reefs, paleoecology, and taphonomy. He was one of the authors of this book, A Sea Without Fish, and that is the title of his talk tonight. Dave.
Okay. Well, thank you all very much for inviting me and for signing on to this program tonight, and we'll just get started. First off, the title is the same as the title of the book that Richard Davis and I published in 2009, and it may be familiar, I hope, to a lot of you. And the talk is going to be derived from parts of that book, but we won't try to cover everything. So you still got to read the book. So this is the cover of our book is part of a let me see, okay, the next frame is the enlarged painting on which the cover is based. It was done by a man named John Agnew, who's an artist in Cincinnati, who has done a lot of wonderful natural history art, both mostly about living organisms in the wild. And he very nicely did this painting for us. And, you know, I want to start out by saying, well, this is the title of the talk, and it may raise a lot of questions with readers, people interested as to just what do we mean by a sea without fish? I mean, this is kind of an unheard of thing. You know, are there any such places or around, or you know, just what does this mean? What do we mean by the sea without fish? And in the cover image here, we see a scene, colorful scene that John did. It looks like it's very shallow water, and you see creatures there. You see some, well, paleontologists and fossil collectors are going to see a lot of familiar things. Nautiloids, they're going to see trilobites, sea stars, crinoids, brachiopods, bryozoans. There's a jellyfish up in the corner. You're not going to see any actual fish. So this is a depiction of the sea without fish. And let's go on here. And here I've kind of written out what will be kind of the plan for the talk and the questions that we want to answer. Okay, so we know that the Earth's oceans have existed millions of years ago, and we're not going to go into all that kind of background evidence. But the question here to start is, have there always been fish and other creatures living in the oceans as they do now? What were they like? How has the life in the oceans changed over time, which we're talking about hundreds of millions of years? And in North America, we have an incredible sedimentary rock record that spans the entire Phanerozoic, and of course goes back into the Precambrian, that contain an abundant fossil record, at least through the Phanerozoic, as a record of past life in the seas that covered much of the North American continent when sea level was much higher than it is now. And we're going to focus on the Cincinnati Arch region of Ohio, Indiana, and Kentucky, the tri-state region we call it here. In particular, has bedrock which is exposed at the surface in this region, which is loaded with abundant fossils. It amounts to about 800 feet of limestone and shale that make up what is called the Cincinnatian series. And these strata are dated, as most of you probably understand, as late Ordovician in age, making them about 440 million years old. And we won't go in too much into the absolute dating story here, but we're going to understand that these are really, really old, some of the oldest known evidence of life in past seas to be found anywhere on Earth. We're really incredibly fortunate in this area to have these right under our feet, literally. So in this talk, we will learn what the fossils tell us about the life of what we call the Cincinnatian Sea, and compare it to the life in modern seas. Major question to try to answer in the time I have is, how has the ecology of the seas changed over time? So there's a focus here on ecology, the relationship between the organisms and the environment of the past. And we're going to start out by looking at different components of the fossil abundance that we have here in Cincinnati. And we start out actually with microfossils. And we're also going to proceed in a kind of an ecological fashion here, because when we look at microfossils in the local rocks, we find evidence of what constituted the plankton of the time of the Cincinnatian. And here's a figure that most of these figures are in our book, but this was done up by Robert Riding in a 2009 paper, and very interesting paper, where here he's plotted on a time scale from the oldest at the left and then to the right up to the present of the components of the marine phytoplankton that are known over time. And this has been something that's interested me for a long time, as to just what the plankton was like. And I think we'll see why I might be so interested in the plankton in the waters that existed. But we're going to see that it's quite an interesting story for the whole Paleozoic, and especially for the Ordovician back here, very early in the Paleozoic. And the plot here is showing the number of species known as fossils of these different groups. So we look at the Paleozoic record of plankton, plant plankton, microalgae in particular. And we have what are called acrotarchs. And that is the only component of the phytoplankton, really, that is well known in the fossil record, not necessarily the entire constituents of the plankton. There may have been things that weren't preserved. You have to remember this, but what was preserved? Well, acrotarchs are organic walled microfossils. Size here, I mean, these things are really, really, really tiny. And their extraction is highly specialized. I don't do it. I've never done it. But rocks are acidized with hydrofluoric acid. These come out of shales. These are found in shales, and the very earliest ones are 1.7 billion years old in the marine sedimentary record. And there were very, very few of them. In the late part of the Proterozoic, there began to be some diversity showing up. And these were the only things, and these have many different forms. They look like little burrs or something. These are magnified in our book some 200 and some times or more even on the page. These are very specialized forms to study. But in the Paleozoic, their diversity became quite high. The different kinds of these things at what are termed to be species. And these were very abundant and thought to be major components of the phytoplankton. What are acritarchs? The name actually means unknown, unknown kind of mystery fossils, but they may be forms related to the dinoflagellates, which come along later. Well, okay, later in the Paleozoic, they decline, they crash, they really crash. But we're interested here in the Cincinnatian, back here, the Ordovician, in this part of the record, when they were very diverse. So they were supposedly a lot of the phytoplankton that was there. So they were undergoing photosynthesis, and they were the major stuff that's in the plankton. Other more familiar phytoplankton groups up here, like the dinoflagellates, you see their diversity is shown here in this plot. Well, they don't really come into the fossil record until after the Paleozoic. And so they're very diverse, and they fluctuate a lot. And today, they're still important components of the marine phytoplankton. Coccolithophores, of course. These are a type of green algae that have a calcareous test. Also part of the modern phytoplankton, and the diatoms. So these components, which most people know to be in the plankton today, were not part of the Paleozoic plankton. So most of the Paleozoic record had none of the things in it that are feeding a lot of the creatures in the sea today at the micro scale, okay? So this is what we start with. What was the base of the food chain, the food pyramid? What did these things look like? All right, here from our book is a plate in which all A through F, the upper half, are all microfossils. The acritarchs here, and down here, a group called echinoderms, which are equally mysterious. These are also organic microfossils. All these things are extracted by hydrofluoric acid out of either shales or limestones, and they're extremely tiny. The echinoderms are kind of flask shaped, kind of like little test tubes or flasks, and are very, very diverse. Both of these things occur in the Cincinnatian. Workers at Ohio State, led by, well, let's see, don't get to Stig Bergstrom yet. People at Ohio State have studied these a lot, and their students have extracted these all out of the Cincinnati and Ordovician rocks and have published on them and described them. It may be then again that the acritarchs are the beginnings of things that became true dinoflagellates later on. The echinoderms, what were they really? They may have actually been protozoans. Well, that would certainly take them out of the class of phytoplankton. They might have actually been single-celled organisms that constructed these little organic tubes in which they lived, and they were part of the plankton. But there's still a lot of mystery in both of these groups as to what they actually were, but they were present too. But down here in the lower half, we have larger forms that certainly were not plankton, they were bottom dwelling. So these were what we call benthic algae, macroalgae, meaning they're larger. You can see my scale here in millimeters is in a lot of the pictures. But these two, H and I, this is about golf ball sized. This is a very interesting thing that has been related. Both these types of things have been related to what are called dasyclads. And these are a major green algae group. They're basically, call them seaweeds if you like, but they were, some of them were calcareous, and the dasyclads existed at the present time. So we've got these things present. and they were living on the bottom, but they're not very, very common, but they could have been a source of food for different larger animal life. Moving on up into larger things, what about sponges? Well, we certainly had sponges in the Cincinnatian assemblage. There's a number of different forms. This is the largest, a group called stromatoporoids. And these are very neat because they were not really spongy, at least in the nature of their basal skeleton, which was calcium carbonate, and forming very, very fine laminations here, microscopically shown, and sometimes forming large domal masses, or there are even some forms that were like stalagmites, basically pillars growing up from the seafloor in the Ordovician. But these are now understood to have been sponges. And one of the key characters making them sponges have been these structures, these root-like, radiating, branching kind of grooves with a central convergence here on lumps that they formed under them. These are called asteroizi, and these are very similar to what are called excurrent canal systems on living sponges, and these exist back in the Cincinnatian, but they're pretty restricted. These got very large. We just drove at 70 miles an hour past a lot of them on I-75 down near Lexington, in the Lexington limestone, which is middle Ordovician, and those have quite a size, a meter or so wide, and very, very a lot of calcification, not forming true reefs, but kind of a reef-like setting. Moving on, we do have cnidaria, which include the corals. Of course, the living cnidarians are very important of the seas, of great importance. Jellyfish, sea anemones are the closest relatives of these corals. There are the jellyfish, the soft corals, the stony corals, the reef building corals are all cnidarians. And in the Cincinnatian, yes, we've got these forms, which everybody probably is familiar with, the rugose corals, the horn corals, which were present in the Paleozoic and can be found in the Cincinnatian, but only in certain horizons. Reconstruction in life shows that they had an anemone-like polyp that was housed in this coral corallum, which is the term, and they either laid on their sides on the bottom, or some might have been partly buried in and stood up like this, and they are thought to have had a ring of tentacles surrounding a mouth by which they fed on things that they captured, but not feeding on plankton, probably on larger things. What were they? I wonder what these things were feeding on. We also have the related tabulate corals, honeycomb corals in the Cincinnatian, and these are colonial. The key thing about the tabulates is that they were colonial corals. Each element here housed an individual polyp, and these are recognized by having radiating septa. They're also tabulates because in the cross section we see the horizontal kind of ladder-like appearance of individual corallites, as each of these is called, as it grew from the ancestral colony through time, and these budded, multiplied, and each of these tabulae supported the soft polyp as the thing grew. And these also make pretty large colonies in certain parts of our Cincinnatian record. So there were corals, but were they reefs? No. I don't think we're going to have time for this to develop this. You might want to question me about this, but the Cincinnatian Sea was not a big place for actual coral reefs to form. This intrigued me a lot. In Madison, Indiana, in a park, a local park, you can find this shed that is actually built of these colonial corals. You can see the hemispherical or spherical shapes here that form the building blocks of this neat little tool shed. These were collected from streams and road cuts near Madison out of the Cincinnatian, from which they are eroded. But they're coming from pretty restricted zones up in the Richmondian, the upper part of the Cincinnatian. And of course, here is a living modern coral, a form called tubastria, a tube coral that you can find living in the Caribbean. But the rugose corals, the horn corals, are extinct, so are the tabulates. They became extinct at the end of the Paleozoic, but they were already present back in our Ordovician time. And here we see extended polyps with their tentacles. These forms extend them mostly at night. In the daytime, they pull them back, the polyp back into the corallite, tuck it right down in there, and they look like this during the daytime. So, but these again are actually scleractinian corals. They are not. They have a different structure. They are not the same group evolutionarily as these Paleozoic forms. But now, of course, we get to the bryozoans. We get to another colonial group of organisms that's extremely abundant in the Cincinnatian. And anyone who has fossil collected in the Cincinnatian knows a lot about bryozoans, or maybe more than they would like to know about. But they are a major component of the fossil fauna here. Entire limestones are formed of their skeletal fragments. So the bryozoans are big rock formers in the Cincinnatian. They, in the ecology of the sea of the Ordovician, they were forms that gave us a lot of the framework of the habitat, this was of major importance as providing a lot of complexity of the bottom and a lot of living space for many other creatures, a lot of cracks and crannies, great diversity of forms, a lot of branching forms. A lot of these resemble very closely many living bryozoans. These are entire colonies that you can find. And the little scale here in each of these four is about 2 centimeters. But this one, look at this one. This is a remarkable thing. This is a kind of a foliose or platy, however you want to describe it, growth form. And this entire thing was excavated out by Ron Fine, a member of the Dry Dredgers, who is an amazing, interesting collector, who has collected these in situ and reassembled them from their pieces. And this one is 65 centimeters wide, really large. And of course, wouldn't be the full size of this. So this is what I mean by, making a framework for habitat. And in amongst these things, we find lots of fossils of other things that lived in with these. These were the nooks and crannies. These were like virtually like corals. I mean, these forms up here were massive, very, very the size of a bowling ball. Here's a fascinating thing. It was an incrustation over a large nautiloid cephalopod that grew across a shell lying on the seafloor. And this is a big, thick, these are the trepistomes. All these belong to the trepistome bryozoans, which are extinct, but we have living counterparts. And here's one of my favorite forms, a living bryozoan, not phylogenetically related to the Ordovician extinct forms, but still showing you the same kind of growth habit. This little colony here would fit in the palm of your hand. I took this picture in the cold waters of the Pacific Northwest. And it's a cyclostomate type of bryozoan, which is found in the fossil record, but not in the Ordovician, came along later. But there's a lot of evolutionary convergence in the colony forms that we get. So we can see a lot of similarity to the forms that existed in the Cincinnatian. Here's a little bit on the what we call the soft anatomy of a bryozoan. Some of them don't have any calcareous skeleton, but these, in all these forms, there's a kind of a tube into which the organism, each member of the colony, called it's not a true polyp, but it's like that. It has a mouth, tentacles that extend, and then a gut leading down into the interior body where all the soft parts, the rest of the soft parts are located. And the food, suspended food, because these are filter feeders, like the sponges we've already seen, that capture their food, take it in, and digest, and then spit out their waste material here. So very important feeding type in the Cincinnati, of course, is that of the filter feeder or suspension feeder. So these would have been feeding on plankton or organic particles in the water derived from that plankton or other sorts. And we've got scads of species of bryozoans. But we've also got brachiopods galore. And I'm sure all you collectors know what brachiopods are. And here's 2 plates from our book showing some of the diversity of the brachiopods in the Cincinnatian. Brachiopods are not molluscs, but yet they have bivalve shells, 2 valves. These forms are all the forms we call articulates, which had a hinged connection between the 2 valves, also made more secure by teeth, cardinal teeth that fit into sockets, and the shells open and closed with the aid of musculature, which we can see beautifully leaving a record here in the muscle scars. And also a lot of these were forms that attached by means of a pedicle, a fleshy stalk, which is found in living brachiopods of the most common types. The articulate brachs are still living today in seas. So they form an interesting analog that we can study to see what the Paleozoic forms might have been like. But this form would have had a functional pedicle. Some of these guys here maybe also a functional pedicle through an opening, but some of these had a pedicle opening that was extremely small or basically closed off in the adult stages. And so they might have been just free lying on the bottom, not attached. A very, very common form of brachiopod in the Cincinnati, and of course, is represented by this type here, the genus Rafinescena. And these belong to the thin-shelled forms that had the 2 valves that kind of nested together like 2 saucers. And each valve being kind of a concavo-convex form. And here we see the same species probably with the convex valve showing here next to one that is flipped over and shows the concave brachial valve. And here we see, this is the interior of this valve here where we see the muscle scars and such. And this is the interior of the pedicle valve, which has the big pedicle opening and really big muscle scars. These formed shell pavements, which is probably a very important component of the bottom structure along with the bryozoans, sometimes covering entire beds like this, oftentimes disturbed by storms, turned up onto edgewise beds like this. And these type of brachiopods are very, very popular as substrates to be encrusted by many other forms, which we'll see that are epizoans or epibiotic forms. But these, most of these probably were lying free on the sea floor, on the soft sediments. There's been a lot of controversy about, well, which way was up, which way was, I mean, this is the upper surface of a bed, and you've got you've got articulated valves here. One is concave valve up, this is convex valve up, and notice the encrusters, which we'll talk about shortly, edrioasteroids, okay? So, and here is an interesting one I want you to see of a Rafinescena that is on an upper surface of a limestone bed, okay? And here you see the hinge would be along here. This is the convex upward pedicle valve. And yet around this margin, notice that there is a kind of a groove, which Ben Datillo and I have started to call a moat, a moat. So whether you like that term or not, that's what we have used for this, a moat, which we have found. And, you know, at first blush, when you see this, you think, oh, well, it's a compactional feature that the shell got compressed by being buried, perhaps before it was petrified, and that the edge of the valve pushed in like a cookie cutter to make this so-called moat. But I think this specimen has, well, you can see it's got two encrusters right on the edge. And these are the edrioasteroids, which we'll talk more about. These are echinoderms. They live, I mean, they're kind of like barnacles really, but they're no barnacle, that's for sure. And we find them oftentimes growing right along the edge of this type of brachiopod. But the very fact that this one has these beautifully preserved edrios, we call them for short, on the edge of this host shell, that is, it is down in this moat thing. And also these two guys sort of collapsed and slumped partly. I think they lived their lives like this, but after death, they kind of get collapsed. Their innards get decomposed, and the plating sags, and they end up like this. But I think this specimen shows that they were living on the edge of this shell when this structure we call the moat existed. And Ben Datillo and I hypothesized that this moat is formed by the brachiopod itself. It's not a burrow per se, but it's evidence that the creature lived a long time in that position and opened its valve for feeding. And thanks to some really elegant research that Ben Datillo has done, and Ben is a professor at Indiana, Purdue University in Fort Wayne, these things could open up almost 90 degrees. The gape angle on the hinge of this thing can be shown to be about 90 degrees. So we argue that these echinoderms were in life position, and this was the host shell was probably alive, because most of these, almost all the ones that are like this are two valves together. They're not broken apart. Here's a living brachiopod. Brachiopods still live. And this little guy is one that is interesting because the pedicle valve on this one is definitely against the substrate, because these little guys cement the pedicle valve to the underside of stony corals in modern reefs. This is recent, and it looks like a little commode, doesn't it? And this looks like the flap of the toilet seat, doesn't it? But this is the brachial valve that, when they're opened almost 90 degrees, you see the filaments of the feeding structure called the lophophore. And this group here is a group that survives to the present. and is found in the Caribbean. These are the structures by which they take in water, they filter it, and they are filter feeders too. So brachiopods are still living. They are not as diverse as they were, and we certainly don't have forms around like these anymore. We don't have these thin-shelled strophomonates, but they were very, very major components of the Cincinnatian. But we also have another group called inarticulate brachiopods. They not inarticulate in that none of them could speak. They were articulate, but the inarticulates lacked a rigid skeletal connection between the two valves. And here we have forms like lingulids that are practically, if you want to call something a living fossil, this is it. This is almost identical to the shell form of the living lingula brachiopod, which burrows into the sediment. And this is a remarkable specimen from the Cincinnatian that was found like this on the edge, vertical. This is the edge of a bed, and this thing was found there, and it would have had a long pedicle coming out of this end that went down into the sediment, and this could probably open up at the sediment water interface for feeding. So this is a form very, very similar to living forms, or the fossil is, the living forms are very similar to these Ordovician ancestors, and they are lingulids. We also have these little cap-shaped brachiopods that are inarticulates. They were also encrusters, and they too have recent species, which again are not that close evolutionarily, but they're the same major groups of inarticulates are around today, and they live the same way. So we've got those type of brachiopods. Let us go to the molluscs. The molluscs, of course, extremely important in the fossil record through time, and we have many molluscs in the Cincinnatian. So there's a lot of interest in what they were like. Here we look only at clams. A and B here are clams of the Cincinnatian. This pair here, this is a attached valves of a form very much like Middleus, the mussel, modern mussels. These are ancestors of those groups. Here's a form that is again probably preserved in life position, a burrowing type of clam or bivalve, kind of like over here in the diagram, these forms here that were probably burrowing, but not very deeply into the sediment. A lot of, you see, these forms here are burrowers, but they did not burrow very deeply, okay? They probably were able to either use siphons, or their gape would open up enough so that they could pump in water currents and extract organic detritus from it for feeding. Other forms lived, well, C and D are belong not to the true clams, but to an extinct group called rostroconchs. Rostroconchs were kind of pseudo bivalve. They had two shells, but they didn't open and close. They had a firm hinge attachment. There aren't any shown here, but they are found in the Cincinnatian too. So we've got all these different types of clams in the Cincinnatian, and many of these are living very much like clams do today. They live on the surface. They are attached. Probably they had byssal threads, which this guy probably did too, by which mussels attach to things on the seafloor. Others attach within things like bryozoan colonies. There's the bryozoans being a nice place for these to live. Others would be on hard grounds, perhaps. There were clams that did probably live on the surface and might have used their siphons to collect organic food material from the sediment, surface feeders. But others did burrow in, but not very deeply, okay? You've got shallow burrowers. These would be what you call infaunal. These up here, living right on the bottom, were called epifaunal. So many of these molluscan life habits were established in the Ordovician, as demonstrated by the incredible contributions by the late John Pagetta, who was a good friend of mine and a tremendous researcher on the Ordovician bivalves, clams. And he with Runigar described the rostroconchs as a separate class of mollusks, and they are extinct, okay? So now, guess what? We're going to jump into my favorite group, the echinoderms, the crinoids, which most of you probably know from your fossil collecting. And we've got a rich record of crinoids in the Cincinnatian. Sometimes they're incredibly beautifully preserved. This specimen here is in the Smithsonian. It was illustrated in 1873 by Meek, one of our pioneer paleontologists of Ohio. Just an amazing complete specimen of a stemmed crinoid. And here is a reconstruction, again made by John Agnew, of a familiar Ordovician form, Glyptocrinus. And here the form is one that we know attached by wrapping its stalk around things like bryozoans. The position of this is derived from our studies of living crinoids, stalked crinoids. And the next picture is an amazing scene that is still available to be visited and observed and studied in really deep water in places like the Straits of Florida. This site is basically between the Bahamas and the east coast of Florida and Miami, Fort Lauderdale, that coast. This is right off, this site is right off Grand Bahama Island in over 400 meters of water. And a good colleague and friend of mine, Chuck Messing, has gone down in submersibles. You cannot free dive or scuba dive. You certainly can't scuba dive. If you're a free diving, you're a very specialized diver. I don't know whether any diving can be done by free divers at that depth. Certainly not this kid. But stalked crinoids live down there today. Now, these are, again, evolutionarily very advanced compared to anything, from the Cincinnatian, which are all extinct. But it was from these that we learned how these things live, that they stand up above the bottom up to 1 meter high. They form what we call parabolic filtration fans. And they bend the arms, they tilt their crown over, they bend the arms back into the current. So the current is going from the bottom toward the top of the frame. You can see ripple marks in the sand here. These are stuck on a hard ground underneath with the attachments there, and they spread out into this fan that's like a radar screen, like a catcher's mitt, however you want to describe it. And they intercept the current going through, and this efficiently captures the flow and enables them to be more efficient suspension feeders. And this you see in the shallow water feather stars that don't have stalks, and we see it in these forms as well. So this is a look back into the past, almost identical forms to forms that existed in the Mesozoic, but certainly not in the Paleozoic. Here's one of these forms, a Cincinnatian crinoid here with its calyx. Its stalk would come out here, its arms here. And here we've got a three phylum trifecta here. We've got a crinoid with a snail, a gastropod, right on the top of its crown, possibly feeding on fecal matter from the crinoid as a host. This is a cyclonema, very, very common in the Cincinnatian and very often in this association. The little guys on the shell of the cyclonema are cornulites. They are little tubular things. They're not corals, but they're kind of like little encrusting corals might be. But they were also a form that probably was a filter feeder as well, that we get as, this is where we have evidence of interactions, probably definitely occurred during life of these species, biotic interactions. And these are some of the very oldest known out of the entire fossil record in the Cincinnatian, the association between the clam and the crinoid as some kind of a host. Some of the clams, some of the snails, gastropods, were actually predatory on the crinoid host, but maybe in the Ordovician, it's hard to say that these snails were not eating the fecal matter of the crinoid, which came out on their surface of their crown like this, okay? So then we've got other echinoderms in the Cincinnatian that are very interesting. The echinoderms, the name kind of means seat stars. Now, these do not have a separate stalk. Instead, they're kind of barnacle-like, as I showed you previously. They form sort of a blister-like or barnacle-like body form that attaches onto something like a brachiopod shell, or it attaches onto just a substrate, a hard ground, or a bivalve or something like that, or even the stem of perhaps a living crinoid. This guy's kind of wrapped around the stem of the crinoid. And these echinoderms are extinct. They went out in the Permian, finally. But in the Cincinnatian, this slide got kind of messed up here. It wasn't supposed to be this large, but this shows the detail of the surface of one of these. They had 5 food grooves that converged on this, what was thought to be the central mouth under these plates. And it's quite likely that the plates, boy, I'm jumping all over here. The plates lining the food groove could open up as kind of zipper-like, and it is thought that they probably possessed tube feet, like living echinoderms, by which they captured food and channeled it to the central mouth. So these were filter feeders also. Their waste came out through this opening here, kind of a valve-like anal opening. And these are found in the Cincinnatian and are especially found on these shell pavements. Okay, now we get to some big favorites for all fossil collectors, trilobites. We've got lots of nice trilobites in the Cincinnati. I can see I'm over 45 minutes now, but we're going to get close to the end of this pretty quickly. Trilobites are, I call, a hands down favorite of collectors, of course. How did they live? What was their ecology? What was their feeding habits? We get them enrolled, the flexicolaminate, or we get them prone. We get them beautifully preserved like this, or we get lots of fragments of the trilobites. We also get incredibly rare and spectacular specimens like this specimen here collected by Dan Cooper and prepared. Alolicus is kind of a not really spine, well, kind of spiny, but a form that even shows what could be preserved color markings. What actual color it was in life or what function it served, we don't know. But this specimen has a different kind of marginal pigmentation that somehow got preserved. It's an amazing fossil. And then we have the giant fossil of the trilobites in the Cincinnatian, the Isotylus. which has been named the state fossil of Ohio. Of course, we've got to have that. This specimen itself measures 37 centimeters and is in the collections of the Cincinnati Museum of Natural History. And these are found in the Cincinnatian. They were very large. It is quite possible that the isotylus and some of the other trilobites were actually predatory. And here is a trace fossil that is a burrow formed by isotylus. We know this to be true because of the size, the great size of this. It matches some of the isotylus that we get preserved as body fossils. This is a hyporelief. It's a trace fossil that's showing on the sole or the bottom of a bed. And it's a cast really of its burrow, which somehow even miraculously was preserved. And even more miraculously, I found this one. This is the best darn trace fossil I ever picked up. Was at a site in Cincinnati, which produced a great many beautiful trace fossils. Richard Osgood, a PhD at Cincinnati under Ken Castor, studied these and became a great pioneer in the study of Cincinnatian trace fossils. It has got traces of the burrowing, the legs of the trilobite, impressions of the dorsal shield, the tail shield, the margins, the pleury here, and the cephalon, again, in kind of a cookie cutter fashion, left these traces. And then most amazingly is this burrow, which looks very much to me as if it was right up by where the mouth of the trilobite would have been. And we hypothesized in a paper that did get published that this may be the evidence of a trilobite killed in the act of going after its prey, a worm perhaps. This is a worm burrow. This is not the only case of this to be known from the fossil record. Some have been found in the Cambrian, they're found in the Silurian, in the formations down in Kentucky. Another thing you want to understand about trilobites is that this plate called a hypostome, which was associated with the mouth on the underside, is quite large in isotylus, and it was forked like this. The mouth probably was located here. And these types of hypostomes were attached to the head shield underneath its front margin. And study of these has indicated that they might have supported different kinds of food habits. I kind of liken this thing to a cutting board, that the trilobite could have held on to some of its food, small organisms that it captured, and used its appendages against this as kind of like a cutting board to chew, macerate the prey, and then move it into the mouth here. And so that's the hypothesis, but it's being revealed that a lot of these trilobites, even the Flexicolumides, might have been predators. Other predators we get to include the eurypterids found in the Cincinnatian, much better known from the Silurian above, of course, but in the Cincinnatian, an incredible thing was found here, the Megalograptus, this guy reaching lengths of over 0.5 meter, and some most incredible specimen that became the type that Ken Castor and Eric Waring described, that was found by a University of Cincinnati student working a summer job along the road cuts, found these specimens, which are in the museum center's collections, the type material. And this guy was a very important type of early eurypterid that was had. And so this would have been a swimmer, not a fish, but probably an active mobile predator, a mega predator of the Ordovician. What about these? More echinoderms, starfish, sea stars, okay? Look at this thing. This is one side of the fossil. This is flipped over, and it's the same fossil from the upper side of the starfish. It looks like a starfish you picked up at the shore today and brought home and dried out and smelled bad, but it doesn't smell bad anymore. But it's a holotype in the Harvard Museum. And here are other forms that are from the Cincinnatian. The most incredible thing is this right here. This is the fossil of a Cincinnatian sea star with its arms holding a clam in the predatory mode of starfish, even today, caught in the act of wrapping and opening up a clam for feeding. So these were predators back then. Here's a form with real flexible, kind of strap-like arms. Here are other forms that look an awful lot like a lot of modern predatory starfish. So these were predators then. We've got, of course, the cephalopods, which participants tonight, like John Catalani, know very, very well. Cephalopods, the nautiloids in the Cincinnatian. We've got the orthoconic straight-shelled forms. We've got coiled forms, look just like a modern nautilus, and we're just about the same size. And we've got these odd certiconic or slightly curved forms that were in the Cincinnatian, particularly in the upper Cincinnatian. The Richmondian stage saw an invasion from elsewhere of diverse predators belonging to the nautiloids. A man who received his PhD at Cincinnati in 1942 was Rousseau Flower, a very interesting character in paleontology, studied nautiloids. And he came up with the idea that these things had to counterbalance their long conical shells by depositing extra calcium carbonate within the chambers that had been early formed in the growth of the animal, and that enabled them to keep themselves counterbalanced, whereas the chambers are otherwise probably being filled with gas, the way that the empty chambers of the Nautilus are. And if they hadn't had that, they probably couldn't have lived to this horizontal position. They would have been bottom heavy. They would have had their heads down all the time, and they wouldn't have been very happy, according to Rousseau's comical reconstruction here. So they got an attitude with this calcification that enabled them to be, quote, happy. They probably didn't smile like that, of course. So these were predators too, and they were mobile. They were planktonic. All right, now lastly, we get to things that might have actually been vertebrates. And there we go back to the microfossils. We've got loads of conodonts in the Cincinnatian. These also studied a whole lot at Ohio State by Professor Stig Bergstrom, Professor Walt Sweet, and many of their students worked on Cincinnatian conodonts, which are extracted from the limestones by acetic acid solution. These are beautiful images provided to us by Stig Bergstrom himself. These are scanning electron micrographs. On a page, these little guys are magnified over 200 times to get on the page. So I don't have the scale right here, but they were real tiny. They are now thought to be actual vertebrates, water conodonts. Were they fish? No. The animal that bore all these things, presumably like teeth somehow, has been discovered from the Carboniferous. Exactly how it lived, I'm not sure that's completely resolved. The conodon animal was probably free swimming, but these things were not the fish that we know today. They were very, very small, but they were prevalent in the Cincinnatian Sea. Are there any true fish in the Ordovician time? Yes. Okay. So the title of our book applies mainly to the Cincinnatian in the tri-state region. It can't be applied globally because here we have a reconstruction of a jawless fish from the Ordovician in Colorado, from the Harding sandstone, very, very famous fossil, which has these dermal plates, which are the forerunners of the bony fish skeleton, but the creature had no internal fish bone skeleton. This was a stracoderm, a bottom feeder that was around in the Ordovician. Other material has been found in Bolivia, other parts of the world. They were present in the Ordovician. So not found in the Cincinnati Arch region yet. When they are, I guess it's time for a new edition of the book with a different title. But the Cincinnatian fauna, when it's put together, and I'm going to have to wind this up, but people have tried to reconstruct what the whole assemblage looked like in dioramas that were first made back in the 1950s. So we can see a lot of these life habits as they were understood then. But now we have some changes we can make. This is a beautiful artistic reconstruction done by Annie Castor and Elizabeth Dalvey, who was an illustrator in our department and the museum. And it shows that we had bottom dwellers, the trilobites, the brachiopods, the bryozoa of different kinds, here bryozoa. There were things swimming around in the water, like the nautiloids, like the graptolites. I haven't even talked about graptolites. And the crinoids were extending up into the water column. And here, the old diorama that was based on knowledge from the mid 20th century, it still exists at the museum and was restored and exhibited recently at the Museum Center, which with comparisons of how these things were regarded in those days. And then some knowledge here about how we now regard the life habits of things like, say, crinoids. They didn't look just like flowers growing up from the bottom. They looked like the pictures I showed you from the deep sea. It's a lot of these graptolites probably didn't float around with floats on them like that, but some of them were probably pelagic. Others lived on the bottom, and many things of the science has changed how these would be made today. Okay, and the way we do this now, this is going to be the windup here, is that a concept called the guild has been used to try to put together all the creatures we find in a fossil assemblage in terms of a food web. You know what food webs, food pyramids are? Well, a good friend of mine, Richard Bombach, came up with the idea of the guild to be applied to these fossil assemblages. A guild, this has been defined by ecologists as a group of species that utilize the same resources, food, space, and so forth, in a similar way. So they will have the same sort of feeding preferences, perhaps using different methods, perhaps similar methods, and forming a guild. The concept kind of relates to the idea of the guild as established back in the Middle Ages. And the way we assign guilds is based on parameters like this. Where does the creature live? How does it occupy space? Okay, that you can have an axis here. Do the animals live on the bottom, on the surface, moving around as epifauna? Do they burrow into it, either shallow or deep? Do they burrow actively or passively? Or are they swimmers? Do they swim as the nekton do, or do they drift passively like as the plankton do? So that defines your spatial component. The food source component is what are they getting as food and where are they getting it? Are they getting suspended? We saw that a lot of the Cincinnatian fauna were filter feeders, suspension feeders. So they were getting suspended matter, plankton, detritus out of the water mass here. And or were they feeding on the bottom? Were they getting detritus, organic matter on the bottom, or were they predators? Were they catching things right on the bottom by moving around? The last component, the bowplon, refers to different characteristics of individual forms, their life history, their growth, and their physiology. And these things enable you to locate different species in gills in different parts of what we term ecospace. All right. Now, to wind up here, something that bears some examination, but we have used this concept to place our Cincinnatian creatures into what are termed megagills. Megagills are big, sort of broad gills that are defined by the living position on the bottom, in the bottom sediment, burrowing, or in the water mass above it. So either infauna, epifauna, or pelagic. And then we define the food source like this, and the feeding type. Filter feeders or suspension feeders, plant feeders, herbivores, meat eaters, carnivores. And of course, everything is based on the primary producers. And so this is something that we did, and some other workers have also done for the Ordovician, is to try to fill in a grid like this with things that we know from the fossil record, how they lived. Everything I've just told you gets summed up in diagrams like this, and this is the result. And the question to look here at is, what are the empty cells, or which would represent broadly ecological niches that existed in the Ordovician? Well, there's empty cells here among the infauna, the burrowers, the deposit feeders weren't around. They either were shallow burrowers or deep burrowers, active burrowers. And we didn't have much deep burrowing by clams or bivalves in the Cincinnatian. Most of the burrows we find are pretty shallow. Deposit feeders were some of the clams, as we said, also worms. We really didn't say anything about those. And some carnivores were probably worms, as they are today. But look at the cells that are really heavily occupied. They're the filter feeders, the suspension feeders, all these groups here that we talked about. Some of them are mobile, moving around, trilobites, sea stars, snails, other groups. And some of them might have been eating plant matter like the seaweeds, the snails, and their relatives. Some of them could have been predators. What about the water column? Pretty empty. Only one known group of surely more things must have been at the base of that food chain, and we just don't know much about those. But there were things, predators like the cephalopods, the eurypterids, maybe some of the trilobites. The trilobites, some of them were thought to be free swimmers or pelagic forms. Conodonts may be down here too. So this is what we can do to make a Ordovician version of this, which is done for the recent marine environment. And you just look at what you get for a recent shallow marine megagill reconstruction. You get most of the cells filled. Well, you don't get deep, you don't get things past just digging in the sediment and sitting there and feeding. They're not going to get much. They're certainly not going to capture much if they're predators. So these are empty, and they're basically the only ones that are pretty empty. evolution has populated all these different cells of such a diagram. And so we can see that the Ordovician was nothing like this modern shallow sea that of course is loaded with bony fish. These didn't come along until starting in the Silurian, the Devonian, of course, and the later Paleozoic. And the fish have made a huge difference in the ecology of the shallow seas. And these were not around in the Ordovician in any form like this, okay? And we can take a slab from the Cincinnatian, and I'll tell you that on this one slab, there were 5 phyla: brachiopods, molluscs, clams, trilobites, that's 3. There's crinoid stems on here, very small, but they're there. So echinoderms were another one. And oh yeah, bryozoans, of course, they have to be there. There's the bryozoans. So 5 different phyla on a single slab like this represents a typical diversity. That's it, folks. Sorry if it went a little over. I'd be happy to entertain any questions, or if you want to email me with any questions, I'd be very happy to answer them in due time. So thank you very much for your kind attention. And
Thank you, Dave.
I hope we understand a little bit more about the sea without fish and why we call it that. Okay.
If you're open to it, Dave, we probably have a bunch of questions.
Okay, well, fire away and I'll do the best I can.
Dave, I've got one little comment.
Yeah.
For cephalopods, at least we found in the Mohawkian that you could actually place cephalopods in 3 additional places on the gill.
Mhm.
Some of the brevicones floated and were planktonic filter feeders.
Yep.
Some of them, the small ones on the ocean floor, were grazers with their radula grazing on algae.
Yeah.
And there were probably some that were actually processing the mud, like some of the gastropods did.
Right.
Kind of a wide variety because of like you were talking about the bow plan, there's a wide variety of cephalopod bow plans.
Sure.
Although they, obviously they're all similar with the septa and the siphuncle and stuff like that.
Yeah. Yeah, well, absolutely. And it suggests that maybe, the cephalopods at that time were filling a lot of niches, if you want to refer to them that way, that the fish have subsequently kind of taken over.
Yeah.
And a lot of cephalopods were doing all these different things. I think that's a really a good comment, John. Thank you very much. And incredible diversity there. And we don't know, the whole story on a lot of this, but this is the kind of picture that we tried to sum up our presentation of the Cincinnatian creatures in the sea without fish. Yeah, thank you. Yeah.
Well, what about the sea scorpion and the jellyfish?
Okay, well, what about the jellyfish that would have been in the Ordovician?
Yeah, and the sea scorpion.
Sea
Sea scorpion, Eurypterids.
Yeah, okay. Well, the jelly, there probably were jellyfish in the Ordovician. I think there are fossils of those. There have been some things in the Cincinnatian first described as jellyfish, a form that looks like concentric circles, markings on the rock, was thought to be a jellyfish, but it's a burrow. So some of these have been interpreted in other ways. And I still don't quite understand what your other example was. See,
Eurypterids, sea scorpions.
Oh, sure, the Eurypterids. Well, yeah, well, they were definitely probably predators. That form Megalograptus had some strange spiny appendages that looked like great rakes. And, I kind of wondered if maybe those things, use those to kind of rake through the sediment to pull up things and then ingest them. They were surely predators, and they were fish-like also. And of course, in the Silurian, they diversified a lot. But the form in the Cincinnatian is one of the earliest eurypterids known in the fossil record, and it's just an amazing thing. Of course, and these were the forerunners of scorpions that made it out of the sea and onto land, leading to things like the spiders and terrestrial scorpions. Sure, but you know, we just don't have any fish. We don't have any plates of these bony, these ostracoderms, these earliest known fish that have been found in places like Colorado. Those don't show up as microfossils here. They just might. There's always new things to be discovered, you know, and a single fossil can
Dave, I was up at Colorado at the Springs,
yeah,
and that last road cut as you go up the hill to Royal Gorge,
yeah.
you can find some of the phosphatic that are in red beds. Do you think possibly that the fish at that time could have been fresh water?
Well, I think that Colorado occurrence has been studied and interpreted as possibly freshwater or nearshore marine. Yeah, definitely. So,
Okay.
you know, that may, you know, a different environment may have been the place where true fish really got going, and that was present in the Ordovician at that time. Yeah, sure, yeah.
I have a question on the brachiopods.
Sure.
I've seen very little, evidence of the predatory holes in many of the fossils of brachiopods. Why do you think there's such a, and maybe it's just that I haven't seen them in very many of the images or, fossil images.
Yeah, like boreholes, drill holes.
Right.
Yeah, well, those become a lot more common in younger parts of the fossil record, when you had snails that were capable of drilling, like moon snails, burrowing moon snails that get their prey that way. And in the Cincinnatian, there have been some circular boreholes found in things. I don't have much experience with it, but some people have described these. They do exist. You know, what was doing that boring? Those snails, those cyclonema, they could have had some boring capability using their radula the way other snails eventually did. The cyclonema are often found associated with the crinoids, like the specimen I showed. They are also found as free snails, gastropods. A member of our dry, excuse me, our dry dredger, Steve Felton, who passed away a few years ago, an incredible fossil collector and astute observer of what he found, was very keen on cyclonema, and he was a great specialist on those, and studied the association of cyclonema with the crinoids. And, you know, they probably had habits that were not just confined to attaching onto crinoids, but they're quite common in the Cincinnati. There are other snails. I'm afraid I didn't do justice to the snails, the gastropods. There are some that are more limpet-like. There are some that are like the monoplacophorans that still exist today. They're kind of like a limpet. We even have Ordovician chitons, the valve shell that is formed by the chiton that then has a foot that it crawls on and attaches. Very important in modern faunas. go back to the Cincinnatian. So too do the scaphopods, the tusk shells that are living burrowed in the sediment and suck water in and feed like that. There's a lot of things that I left out that maybe they don't occur in the Cincinnatian, but they certainly could have been part of the story. Getting back to the jellyfish. Well, from other parts of the world, I guess there are Ordovician jellyfish they're definitely undoubted jellyfish. I mean, look at the Mazon Creek biota with lots of jellyfish. Were described by many workers, just beautifully preserved. I mean, jellyfish were around, and they would have been part of the mobile predators or plankton feeders as well. But I think we're kind of stuck with a lack of knowledge of what was the whole base of the food chain. Even those acrotarchs were there, probably abundant, but it wasn't anything like the kind of organic soup of later seawater environments, as I showed you. And I think there's a lot more we need to learn about the evolution and history of the plankton in the sea. I'm interested a lot in this for the Carboniferous too, the Mississippian, and what the sources of food were at that time too. Because I mean, the Paleozoic, what we see in the Cincinnatian becomes very typical of the entire Paleozoic marine fossil record throughout the world. I mean, it's the beginnings of great diversification. We know that Cincinnatian is part of a Ordovician radiation of fossil producing organisms, invertebrates, and this is just the beginning of a big Paleozoic story. But I really think that the Paleozoic marine ecology is really different than what came later. And I think we've got a lot to understand about the nature of the marine ecology of the Paleozoic. And of course, that's factors in, what led to mass extinctions. We have a big extinction at the end of the Ordovician, and then we've got a whole lot more in the Paleozoic. And a lot of these, forms are big components of the marine fauna in the Paleozoic, became extinct ultimately at the end of the Paleozoic and the late Permian extinctions. The crinoids almost go out completely. Things like the Edroasteroids, a lot of these groups are gone. The corals that I showed you are Paleozoic restricted forms, and new things take over. But we can still learn a lot from the living counterparts or the analogous forms, not necessarily the direct descendants of the early faunas, but things that have adopted the same life habits and the same morphology. I think it's, the present remains a key to the past, but the past also informs what we understand about how the present evolved. Okay, so
Dave,
yeah.
I graduated University of Dayton.
Yeah.
84 with a geology degree.
Yeah.
And I was just wondering if you knew any of the professors there.
Oh, sure. Dan Goldman.
Goldman? No, he must not have been there in 84, but Charles Ritter.
Okay, yeah, not too well, but there have been people there that like Goldman work on graptolites, right? And I really gave them really short shrift. Graptolites were both free floating planktonic forms and also bottom dwelling benthic forms, colonial forms. And they too were filter feeders. The graptolite animal is pretty well known. You know, again, another group feeding on some very small prey or food sources, particulate matter or actual living plankton. I mean, what was the zooplankton like? I mean, we've talked about the possibly the phytoplankton, but what was, you know, you talk about the consumer level of the food chain, the food pyramids, and we've jumped right into a lot of large animals, invertebrates, and what about things like modern zooplankton, like the copepod? I mean, all those things didn't come along until much later in time, and yet, you know, you have to realize that a lot of planktonic forms might not have left a good fossil record at all. They could have been soft-bodied, just virtually unpreservable. And we've just got to remember that a lot of, you know, that goes for the jellyfish too. They're just not going to be preserved. You have to keep in mind that a lot of things we see today just are not going to leave a fossil record. We have to understand what the biases of preservation are. And paleontologists have devoted a lot of attention to that over many years and still do. I think it's very important. Yeah. Any other questions?
I have a question on the two things on the trilobite that you showed with the within a burrow.
Yeah.
The first question is, what made you determine that the trilobite was the predatory and not the worm?
The worm. Good, very good question. Well, okay. Well, the worm burrow is located where it is, you know, right where the mouth of the trilobite's mouth was under the front end of its head shield, its cephalon. And, you know, it really looks as if the trilobite was digging down into the sediment, and it cut off the worm in its burrow. That's what it looks like. So
Okay.
we interpreted it that way. And let me see, there was evidence of scratch marks on the worm burrow that were possibly created by the appendages of the trilobite. Because remember that the trilobite appendages are almost never preserved.
Right.
And yet those are the primary equipment they've got to get food and to consume it. And I mean, the isotylus, they're actually specimens that have preserved appendages. And then if you think of another trilobite that's in the Cincinnatian Triarthris, those have been found not here, but from New York State, where the appendages, the soft appendages, are preserved. They were described by Beecher, very famous beds equivalent to the Cope, the Utica shales of New York. And they've studied those with X-ray examination, and have revealed beautiful details of these things. And now we've got micro CT scanning that reveals a lot of soft part anatomy of things that have not been studied. And so, you know, we're getting to know a lot more about trilobite appendages, and especially I talked about those hypostomes. Those are, I think, very instructive structures to examine, to find out what the different forms are, and, you know, how they might have been used by the trilobite. I mean, the hypostome on Flexicolamintes is a little shield-like thing. There's quite a lot of study of the forms and the diversity of trilobite hypostomes that you might want to. I could suggest papers that go into that. And we have a collector in Ohio Steve Brown, a very excellent collector of trilobites, and he has studied the hypostomes in the different trilobites in the Ordovician, and they're very diverse, which seems to point towards different ways in which the trilobites were using them. So, when you get into that bow plan aspect of the ecospace, if you will, that points to great diversity at a lower taxonomic level. The mega guild is putting whole groups into these spaces in the diagram, and it's a pretty coarse analysis, let's say, coarse-grained analysis. Okay.
So the other question I had was on the trace burrow around the trilobite.
Yeah.
And this is just a general, I found myself wondering how many collectors have not realized it was a burrow and removed it thinking it was just matrix. Same with the worm burrow. And even the encrust parts of, the brachiopods. I mean, I probably have removed it thinking it was just matrix. And I wonder how many have had that happen.
I would say it's probably quite frequent. I mean, those structures around the brachiopods, not everybody agrees with Venditillo and me on that. People think was produced by scour, by currents. It's not. I just do not think it's scour. We can basically prove that. I mean, Ditillo has done some elegant research with the brachiopods, where he found evidence that the brachiopods were actually actively burrowing or escaping being buried by a storm. And he found this first in, there's a small brach called Sourbiella that is found in the Ordovician, much smaller than our Raffinescinas, but the same type of thing. And he found Sourbiellas that had been buried, in a sediment movement of some type, and that they showed a trace where they by probably moving the valves. I mean, we think that even the Raffinescinas, if they were covered by a storm produced sediment flow burial, that they could basically clap the valves and get themselves out. They could do escape burrows. And in a paper we published in 2009, we looked at evidence for these modes to have been an actual part of a burrow that had more to it than that. So yeah, I think you're right that happens, and people need to look at the sediment around your fossils, and is there evidence of any life activities, and what it might have been. I mean, the whole trace, I didn't, beyond that, there was nothing else on trace fossils here. But we learned from trace fossils tremendous things about the life habits of the organisms. They are the record of behavior. So feeding is a big thing about this. And that's just absolutely fascinating. I mean, the Cincinnatian, there could be a whole program on the trace fossils. And we had the beginnings of that with Dick Osgood and his mentor, Kenneth Castor, who was studying this stuff back in the 30s. He was studying how trace fossils are formed. Tracks that were thought to be vertebrate amphibian tracks. Castor showed that they were formed by horseshoe crabs crawling. And that was back, he was doing experiments with these horseshoe crabs to find out what kind of tracks they might leave. And by golly, they're very similar to a lot of things that were thought to be small amphibian tracks. No, they weren't. So
What do you suppose this is? I found this near Cincinnati.
oh, Okay, is it a, well, you can find a lot of internal molds of cephalopods. It's probably a body chamber mold. And a lot of those will still have the septa, or if it's the body chamber, it would just be a solid thing like you've got there. And I can't see the details on it.
Okay, yeah, it doesn't, it's just solid. It doesn't have
Yeah,
the
yeah,
fibers. So
yeah,
it's,
yeah,
this probably
yeah.
came from a big cephalopod.
yeah. And I mean, some of those, were huge. We don't, some in the Cincinnatian probably were a meter. There are specimens in the collections and were really big.
Closer.
And, yeah,
Oh, or-
yeah.
Yeah, it's
Yeah, it looks like probably a filling. And then see what could happen is that the actual shell was not preserved and the internal filling might have been petrified, and that's what you end up with. We find those things have got fossils on them like trilobites or other things that probably lived inside an empty shell and got trapped in there, buried, and are preserved. They're pretty amazing too.
Wow, I never would have thought to look inside. Yeah, that's amazing.
Yeah, and I mean, we've got some internal molds of cephalopods that have got entire trilobite, even larger trilobites like Flexicolimenes on them. And so, you probably had the empty shell being a place the trilobite crawled in for shelter and then got buried somehow. Yeah, and so there's all these neat things about life habits that we can interpret from careful collecting and observation. Yeah. Good questions, folks.
Point, I'm gonna end our recording and
Okay.
open up the floor to anything and anything. So if you want to hang around, Dave, for more questions, or if you want to just participate in the conversation from this point, be my guest.
Well, I can hang around. I'll take any more questions that are burning.